Welcome to another lecture by Medical Medics, Immunology, Chapter 3, Innate Immunity. In this lecture, we will dive deeper into the concept of innate immunity, talk about its components, differentiate between physical and chemical barriers, talk about the main cells of innate immunity, soluble components of innate immunity, and pattern recognition in innate immunity. Furthermore, we will briefly mention the inflammatory response, the innate immunity in action. We will differentiate between innate and adaptive immunity and end with a summary. So, innate immunity is the non-specific, immediate defense mechanism that protects the body against pathogens. Key features include that it is non-specific, meaning it responds to all pathogens the same way. It is immediate, so it reacts within minutes to hours. But it does not have memory, so it does not improve with repeated exposure. The components of the innate immunity include physical barriers, so we talk about the skin, mucous membranes, and epithelial layers that prevent pathogen entry. We have chemical barriers, so enzymes like lysozymes in our tears. We also have acidic environments as part of the chemical barriers, like stomach acid, which helps kill pathogens. Furthermore, we have cellular defenses, so immune cells like neutrophils, macrophages, dendritic cells, and natural killer cells. Furthermore, we have soluble factors, so complement proteins and cytokines that enhance pathogen destruction. So the physical and chemical barriers, as mentioned, include, for example, skin, which is a tough, waterproof barrier that blocks most pathogens. We also have our mucosal surfaces, which help trap pathogens in mucus. These surfaces also contain antimicrobial peptides like defensins. Then we have the chemical defenses. Stomach acid, for example, which helps destroy ingested microbes. We have a lysozyme, which is in tears and in our saliva, that helps break down bacterial walls. So this is all part of our immune system. Innate immune system, to be specific. What about the cells, then, of our innate immunity? Well, we've repeated these in our previous introductory lessons, but it's always worth repeating because it is quite high yield. And even though there are few, learning to differentiate between them is always good to repeat. So we have the neutrophils. These are the most abundant immune cells. They help engulf and destroy pathogens through the process of phagocytosis. Then we have the macrophages, also involved in engulfing pathogens and dead cells. They are also antigen-presenting cells, so they present the antigen of the pathogen that has been broken down to our adaptive immune cells. Furthermore, we have dendritic cells. These help capture antigens and transport them to lymph nodes. They are also crucial in linking together our innate and adaptive immunity. And finally, we have natural killer cells. These are mainly involved in destroying virus-infected and abnormal cells. So, soluble components of innate immunity. Here we have cytokines. These are small proteins that regulate immune responses. For example, interleukins and interferons. You will come to learn that there are several types of interleukins and interferons. Now you can think of them as amplifiers of a kind. So to facilitate an immune response, these are players that play a role in amplifying the immune response. We also have chemokines. These attract immune cells to infection sites. Furthermore, we have a complement system. This is a group of proteins that opsonize pathogens, meaning they mark them for destruction. They also create membrane attack complexes to lyse pathogens. So the cytokines and these complement proteins are like messages and ammunition that help our immune cells fight invaders. So then, pattern recognition in innate immunity. So the innate immune system responds quickly by recognizing shared molecular patterns found in pathogens or damaged cells. So we have pattern recognition receptors, or PRRs. These are receptors found on or inside innate immune cells, like in macrophages, dendritic cells, for example, that help detect these molecular patterns. We have pathogen-associated molecular patterns, or PAMPs. So molecules commonly found in microbes, such as lipopolysaccharide, LPS, on, for example, gram-negative bacteria, or flagellin from bacterial flagella. Furthermore, we have damage-associated molecular patterns, DAMPs. So these is molecules released by stressed or dying host cells, such as ATP or nuclear proteins, signaling tissue damage. For example, one example of a PRR are the toll-like receptors, TLRs. And we have a toll-like receptor, TLR4, if memory serves me, that recognizes the lipopolysaccharide on bacteria. So when it recognizes the LPS, it will trigger an inflammatory response. We also have NOD-like receptors. So for example, NOD2 detects bacterial peptidoglycan fragments. Bacterial peptidoglycan fragments inside cells, and this is important in gut immunity. So, to summarize, innate immunity acts as the body's first line of defense by using PRRs, so pattern recognition receptors, to quickly recognize dangerous signals. These receptors don't target specific pathogens like the adaptive immune system does, but instead they detect conserved molecular patterns, so PAMPs, for example, or DAMPs, and that are common across many microbes. And these early signals are essential in launching the immune response, helping to recruit other immune cells, and then help shaping the subsequent adaptive immune response. Let's briefly repeat the inflammatory response. So we have several steps when we talk about inflammation. First, recognition. So immune cells detect pathogens or an injury. Then there is a release of signals. We mentioned cytokines and chemokines that help recruit other immune cells. There will be an increased blood flow to the area, which causes redness and warmth. There will be a recruitment of neutrophils and macrophages. This leads to swelling and pain. And finally, there will be a resolution where the pathogen is cleared and the tissue repairs begin. And so here we also find the key signs of inflammation. We have redness, we have warmth, we have swelling, and we have pain. So let's briefly take a look at innate immunity in action. So example 1, a cut on the skin introduces bacteria. Neutrophils arrive first to engulf the bacteria, while macrophages clean up the debris. So when the skin barrier, which was one of the physical barriers of our innate immune system, when the skin barrier is broken, such as from a cut, for example, or a scrape, bacteria from the environment can easily enter the body. In response to this, the innate immune system activates immediately. Neutrophils, which are the most abundant type of white blood cells, are the first responders. They are rapidly recruited to the site through signals like chemokines and perform phagocytosis, meaning engulfing and killing the invading bacteria. Soon after, the macrophages arrive, and they help clear away dead cells. They help clear away pathogens and debris. And they also secrete pro-inflammatory cytokines, like the interleukins we mentioned. For example, interleukin 1. Another one is tumor necrotic factor alpha. And these help sustain the response. A point of correction, it's actually tumor necrosis factor alpha. But hey. Now, when all of this is happening, this process often then leads to the visible signs of inflammation mentioned in the previous slide. We will see redness, swelling, warmth, and pain. All of these are hallmarks of innate immune activity. Another example, a viral infection triggers interferon production, which inhibits virus replication. So during a viral infection, such as from in the influenza, for example, or SARS-CoV-2, cells detect viral genetic material. And they will then produce type 1 interferons. These interferons act in both autocrine and paracrine fashion. So they bind to receptors on infected and nearby uninfected cells to trigger an antiviral state. This inhibits viral replication and helps contain the spread. Interferons also enhance the activity of natural killer cells and promote antigen presentation. This helps initiate the adaptive immune response. So without this rapid interferon response, viruses would replicate unchecked in the early phases of infection. So, key differences between innate and adaptive immunity. Some of this is repeated because you've probably seen the previous two lectures, but if you haven't, the innate immunity, when we talk about speed, it is usually immediate, meaning minutes. Adaptive immunity can be delayed up to several days. Specificity, so innate immunity is non-specific, it's a general response, and adaptive immunity is highly specific. When we talk about memory, in innate immunity, there is no memory, but there is a long-lasting memory in our adaptive immune system. Key players in our innate immunity are neutrophils and macrophages. And we can add dendritic cells, and we can also add natural killer cells here. When we talk about adaptive immunity, we have our T cells and B cells. So in summary then, innate immunity is the first line of defense, providing immediate and non-specific protection. Components include barriers, immune cells like neutrophils and macrophages, and soluble factors like cytokines and complement proteins. It relies on recognizing common patterns of pathogens to mount a response. That's the end of chapter 3. Continue now to chapter 4.